Hi, Mr. Kensting here. Going to animate transcription translation using this paper chromosome right here, which is a double-stranded molecule of DNA nucleotides, where nitrogen bases A and T are nucleotides A and T are complementary and G and C are complementary. So the very first thing that happens in transcription, which happens in the nucleus of the cell, and so this is going to represent the nucleus right now, is a special enzyme called RNA polymerase is actually going to kind of pop the DNA molecule open a little bit. And what it's going to do is it's going to build an RNA molecule, which is different than the DNA molecule, but similar also. It's going to build something that's complementary to this DNA. So it's going to copy this information. The A matches the T because it's complementary. The U matches the A because it's complementary. The G matches the C because it's complementary. The G matches the C due to complementary, and so on. So the G goes with the C, the A with the T, the U with the A, the C with the G, the U with the A, the U with the A, the G with the C, and the A with the T. Now, at this point, the RNA polymerase is done with transcription. This molecule here is called messenger RNA, but it doesn't stay here in the nucleus. It actually goes out to the cytoplasm. What I'm going to do, instead of moving the RNA, I'm going to show that, kind of animate that this is going to leave the nucleus and going to go out to the cytoplasm. So I'm going to move these off so that the messenger RNA is actually moving to the cytoplasm now. Now this is going to represent the cytoplasm, and this is my RNA, messenger RNA molecule that it is going to aid in the production of a protein by a process called translation. So the very first thing that happens is a ribosome will attach to this RNA molecule, and the ribosome is basically a molecule that actually covers about 2 codons. These triples of letters like AUG represent a word called a codon. So this is AUG is a codon, GGA is a codon, UCU is a codon, and UGA is a codon. When the RNA, or the messenger RNA, is found by a ribosome, what happens is transfer RNA molecules come in carrying amino acids. So this transfer RNA has an anticodon UAC, which is complementary to the codon AUG. And this transfer RNA molecule carries the amino acid methionine. Methionine is the very first amino acid of our protein here. The second amino acid that's going to be brought in by a transfer RNA will end up being glycine, and that's because the next codon is GGA, and the anticodon that matches it is CCU. At this point, the ribosome does a very important thing. It makes a peptide bond between the glycine and the methionine. At that point, the very first transfer RNA that came in, it goes out to go find another methionine, okay, for another ribosome. Meanwhile, the ribosome will shift down 1 codon. Now it's looking for a transfer RNA with an anticodon of AGA, and that comes in carrying serine. And so it locks into position, and the ribosome does the next best thing. It bonds the serine to the glycine with a peptide bond. At that point, this transfer RNA molecule goes out to go find another glycine. The ribosome shifts down 1 more codon, but at this point, this is a stop codon, UGA. The ribosome recognizes that, and what it does is it cuts away this transfer RNA from the amino acid it was carrying, so it'll go out and get another serine. At this point, it detaches from the messenger RNA. The messenger RNA molecule will leave and go find another ribosome to make another protein. These amino acids will often roll up into a ball or form some goofy shape. This is a finished polypeptide, and the very beginning of the methionine here where the stub is sticking out, this is the amino end. The amino end is always the methionine end of a protein. And this right here where you see the notch, that's the carboxyl end of the protein. That's always the last amino acid of the polypeptide. The tail end of that amino acid sticking out is a carboxyl group. That's called the carboxyl end. So this finishes the animation of transcription translation, the whole purpose was to get the message of a DNA molecule that was double-stranded into a sequence of amino acids that represents a protein, which could be your hair, fingernails, blood proteins, antibodies, or any of the other enzymes that make the human body work.